During G.1 trilation, highly coordinated cell movements transform the blastula from a ball of blastomers to a highly organized embryo called a gastrula with 3 distinct layers from which all organs and tissues originate. The 3 layers of the gastrula are collectively called embryonic germ layers, and include the ectoderm, cream, and cream. While presumptive, future, ectoderm cells remain on the exterior of the gastrula, blastomers that will become mesoderm and endoderm migrate internally. The new locations of these cells allow tissues and organs to form in their proper positions in a process called organogenesis. The ectoderm gives rise to the epidermis and components of the nervous system. The endoderm generates the epithelium of the gut and gut derivatives, example liver, pancreas, and the mesoderm forms the circulatory system, muscles, kidneys, gonads, dermis, and skeletal components. At the end of gastrulation, the gastrula is characterized by an inner layer of endoderm, an outer layer of ectoderm, and a middle layer of mesoderm. Understanding how cells differentiate into the 3 germ layers and exhibit specialized movements is one of the major areas of research in developmental biology today. Overview of gastrulation. Gastrulation movements involve the entire embryo. Cell movements of any area in the gastrula are synchronized with movements of other areas of the embryo. Although the pattern of gastrulation can vary amongst different species, the embryos exhibit similar types of basic cell movements as seen in Fig. 7. 1. Invagination, infolding or buckling inward of a sheet of cells, analogous to a finger poking a balloon. Compare with involution and angrization. Example, sea urchin endoderm. Involution, inward migration of an expanding outer sheet of cells so that it spreads back over the basal, internal, surface of the remaining external cells. Compare with invagination and angrization. Example, amphibian mesoderm. Angrization, migration of individual cells into the embryo's interior. Compare with invagination and involution. Example, sea urchin mesoderm. Declamination, splitting of a single shear of cells into 2 separate but parallel sheets. Example, chick and mammalian hypoblast formation. Epiboly, expansion of an outer sheet of cells by mitosis to cover the entire embryo's surface. Movement is analogous to syrup being poured over an orange. Example, ectoderm in sea urchins, runicates, amphibians, fish, and birds. Convergence, migration of cells toward a common point such as the midline of the embryo. Examples, cell migration over the frog dorsal. Blastopore lip, and chick primitive streak formation. Extension, rearrangement and elongation of a cluster of cells to form a thin, stretched cellular group. Analogous to a group of people that form a large crowd while waiting for a bus, but move in single file when boarding it. Example, sea urchin endoderm during archenteron formation, and cell migration over the frog dorsal blastopore lip. Gastrulation in model organisms. His chapter explores the process of gastrulation using several model organisms to demonstrate different gastrulation strategies. Their order of appearance reflects their complexity of gastrulation. Preserved whole mounts are shown unless indicated otherwise. In diagrams of gastrulae, the embryonic germ layers are given specific colors by convention. Ectoderm is blue, mesoderm is red, and endoderm is yellow. This enables easy identification of the germ layers of gastrulae. Echinoderm gastrulation. Sea urchins exhibit a simple type of gastrulation that occurs soon after the blastula hatches from the fertilization envelope, Fig. 7.2, Table A.5. At the onset of gastrulation, primary MCCM hymal cells, derived from micromers, ingress into the blastocele, and later differentiate into mesoderm. The vegetal side of the blastula flattens into a vectal plate that invaginates, forming the anchron, embryonic gut, also called gastrocele. The opening of the archenteron is the HB toperc, which in sea urchins is the site of the future anus. The archenteron elongates by convergent extension, reaches the inner surface of the blastocele wall, and fuses with the ectoderm to form the stomatocum, mouth. The anus marks the future dorsal side, aboral side, and the stomodium forms on the ventral side, oral side. The gastrula next progresses through the prism and plutkus larval stages, Fig. 7. 3 to 7-6, Table A.5. The prism stage does not have brval arm, whereas the pluteus has two pairs, a prominent long ventral pair, anal or dorsal arms, and a short dorsal pair near the mouth, oral or ventral arms. Sperm made of calcite, caco, MgCO3, provide skeletal supports for the larval arms. The spicules are difficult to identify in preserved larvae since they are easily dissolved by commonly used fixation methods. At the prism stage, the spicules begin to lengthen, giving the embryo a prism shape. Further spicule elongation enables the larval arms to grow out in the plurius. The archenteron differ entiates into three regions of the gut: esophagus, stomach, intestine, which appear shaped like the letter J in the pluteus. The plurius is covered with cilia which allow it to swim freely with its arms forward. The pluteus will later metamorphose into a small sea urchin, which grows spines, becomes sedimentary, and resembles the adult form, Fig. 1.9. Starfish. The starfish blastula undergoes gastrulation similar to the sea urchin in that its vegetal side flattens, forming a vegetal plate, and invaginates to form the archenteron, Fig. 7.7, 7-8. The blastopore becomes the site of the anus. The archenteron of the mid-gastrula stage starfish evaginate, expands outward, at one end to form the archenteric vesicle which generates mesenchymal cells, mesoderm, that scatter throughout the blastococle. The archenteron also gives rise to the mesoderm cochlomic AC. Chylomic pouch, which develops into the cochlum, body cavity, and water auriculae cavities of the adult, Figs. 7.9 to 7.12. The chylomic sac will eventually completely separate from the archenteron. The archenteron undergoes additional swellings and constrictions to form the stomodeum, esophagus, stomach, and intestine. By this stage, the embryo is called a hyponaria larva, Fig. 7.7, 7.10, 7.12. Within 36 to 48 hours after fertilization, free-swimming bipinnaria larvae begin to ingest food, Fig. 7.12. After this stage, the bipinnaria metamorphoses into a benthic starfish that takes on the adult form, Fig. 1.10. Nemerteon gastrulation. The blossrula of the milky ribbon worm, Cerebratulus, is covered with cilia that rotate the embryo within the fertilization envelope until it hatches at the gastrula stage, Table A1. The apical tuft is a bundle of long cilia at the animal pole that is connected to the apical organ, apical plate. The animal pole becomes the dorsal side of the adult. The blossrula has a large blastocele and a flattened vegetal plate, which becomes the ventral side of the adult. Gastrulation begins when cells in the middle of the vegetal plate invaginate or ingress into the blastocele, forming the archenteron and blastopore, Fig. 7.13. The archenteron is blind in that it is pouch-shaped with one opening, the blastopore, side of future mouth. As the archenteron elongates, it curls to one side. Angrazion of mesoderm generates mesenchymal cells that scatter in the blastocele. The larval stage is helmet-shaped, and is called a pylodium larva. The ectoderm surrounding the blastopore of the pylodium larva extends past the blasropore to form lateral lappets. Gastrulae and pylodium larvae are free-swimming and rotate along the animal vegetal axis, moving with the apical tuft forward. After about 12 days, the pylodium larva metamorphoses into a juvenile worm that later reaches adult size, Fig. 1.2. Cephalochordate gastrulation. Gastrulation begins in the lancelet, amphioxus, branchiostoma, with the flattening of presumptive endoderm blastomeres at the vegetal pole into an endoderm plate, Figs. 7.14, 7. 15, Table A.6. This plate of cells invaginates and completely eliminates the blastocele. The embryo becomes double-walled and cup-shaped with a large blastopore representing the cup's opening. The outer wall of the gastrula is presumptive ectoderm, and its inner wall of endoderm forms the archenteron. Thickened lips surround the blastopore. Future anus. The dorsal lip contains presumptive notochord cells, chordomesoderm, and the ventral and lateral lips contain precursors of mesoderm. Teleost gastrulation. At the beginning of teleost gastrulation, the blastoderm has an outer epithelial cell layer, enveloping layer, and several layers of deep CCLK. Zebrafish gastrulation begins when the blastoderm has spread by epiboly over slightly more than half of the yolk cell, Fig 716, 717, Table A.7. While the enveloping layer undergoes epiboly, deep cells near the blastoderm margin on the future dorsal side of the embryo involute to form two layers of deep cells, 1, an upper layer of epiblast cells, future ectoderm, that have not involuted, and 2, a deeper layer of involuted hypoblast cells, future endoderm and mesoderm. In some fish, delaminating/engrossing cells also contribute to hypoblast. A germ ring forms that is a distinct thickening of the entire margin, and consists of epiblast and hypoblast. Eventually deep cells from both epiblast and hypoblast converge on the dorsal side and intercalate to form the embryonic shield, embryonic axis. The embryonic shield is considered the fish organizer because it organizes cells to form the body axis of the embryo. It induces embryonic cells to differentiate. Amphibian gastrulation. The pigmented animal hemisphere of amphibian embryos, Ranopapiens, Xenopus laevis, makes cell movements during gastrulation visible as they drastically change the embryo's appearance, Fig 718 to 7.20. Gastrulation begins on the dorsal side in the gray crescent at the marginal WRN, an area surrounding the equator of the embryo where the micromers and macromeres meet. A group of cells, bottle cells, in the dorsal marginal zone constrict at one end, becoming bottle-shaped and invaginate to form the leading edge of the archenteron. This causes a pigment difference on the embryo's surface which becomes visible as the dorsal lip of the blastopore, over which marginal zone cells, future mesoderm, involute. In some frogs, the archenteron roof is initially lined with involuted mesoderm, chordomesoderm, that later becomes covered with endoderm. The archenteron floor is lined with vegetal cells that become endoderm. The blastocele is displaced as the archenteron enlarges. Eventually the blastopore lip widens into a crescent forming lateral and ventral lips as additional marginal zone cells involute. The dorsal lip of the blastopore has been called the frog organizer, Speimann's organizer, since it induces cell fates and organizes cells into specific tissues. It is analogous to the fish embryonic shield. The blastopore is the site of the future anus in amphibians. Exterior cells of the animal hemisphere proliferate and move by epiboly to cover the entire embryo's surface with pigmented micromers. Near the end of gastrulation, the dark brown embryo has a small, light yellow yolk plug, patch of endoderm cells that has not yet been covered by pigmented cells, and is bounded by the lips of the blastopore. Internalization of the yolk plug by epibolic movements of the ectoderm signals the end of gastrulation, Table A.8. Avian gastrulation. Avian gastrulation begins in the embryo's thickened caudal margin, marginal zone or belt, where the area pellucida and area opaca meet. The direction of cell movements during gastrulation is shown in Fig 7.21. Cells migrate into the embryo through the primitive streak, a midline thickening of epiblast located in the caudal two-thirds of the embryo, Fig 7.22 to 7.34. Primitive folds, primitive ridges, flank a midline depression down the primitive streak called the primitive groove. Future endoderm cells are initially located in the epiblast, but later ingress through the primitive streak to populate the hypoblast. Prospective mesoderm cells migrate through the primitive streak and populate the Area between the epiblast and hypoblast. Cells that ingress through the primitive pit, most rostral region of primitive groove, form, 1. A thickened area rostrally called Henson's node. Primitive knot, and 2. The head process which becomes the anterior portion of the notochord. Henson's node is the avian organizer. If experimentally transplanted to an ectopic site, it organizes an entire additional embryo. The primitive streak is first visible from 6 to 10 hours of incubation after egg laying, stage 2, table A.9. It elongates and reaches its full length by 18 hours of incubation, stage 4. The primitive streak then regresses in the cranial region of the embryo as future mesoderm and endoderm cells migrate through it. The primitive streak is visible only in the caudal region of the 33-hour embryo, stage 10, and has regressed completely by 48 hours, stage 13. Mammalian gastrulation. Gastrulation in eutherian mammals is very similar to avian gastrulation despite the lack of yolk. The primitive streak is the site of gastrulation. The bilaminar blastocyst undergoes gastrulation during days 6 to 8 in rodents and day 9 in pigs, tables A10, A11. Gastrulation commences when the primitive streak appears in the caudal end of the epiblast and extends rostrally. At the rostral end of the primitive streak is a mound of epiblast called the primidia node or node, which is the mammalian organizer. As with avian gastrulation, mammalian epiblast cells ingress and involute through the primitive groove and pit into either the space between the epiblast and hypoblast to become future mesoderm, or into the hypoblast to become future endoderm. As gastrulation proceeds, the embryo becomes trilaminar with the germ layers derived from epiblast distributed as follows: ectoderm remains in the epiblast, endoderm replaces the hypoblast, and mesoderm migrates between the epiblast and hypoblast. As gastrulation proceeds, the primitive streak regresses caudally. Pig at about 7 days after fertilization, Carnegie stages 3 to 4, table A11, the inner cell mass of the spherical pig blastocyst has flattened to form the bilaminar embryonic disc consisting of an outer epiblast and inner hypoblast layer similar to the avian embryo, Fig 7.35a, b. Proliferation of the trophoblast cells starting on day 8 causes the blastocyst to undergo a dramatic shape change from spherical to tubular to filamentous by day 12, Fig 7.35c. The shape change increases the embryo's surface area for exchange of materials between the embryo and uterus. By 10 to 12 days, Carnegie stage 7, the thread-like embryo is up to a meter long, and the embryonic disc is located about midway along the length of the thread, Fig 7.35c. While the blastocyst undergoes elongation, the primitive streak begins to develop in the caudal region of the embryonic disc about 9 days after fertilization, Carnegie stage 6. The primitive node is located at the rostral end of the primitive streak, Fig 7.35d. The primitive streak of the 12-day-old pig embryo, Carnegie stage 7, is comparable to that of the 1-8 to 1-9-hour chick embryo, stage 4. Mouse at about 4.5 days after fertilization, Thiel ER stage 6, table A10. The inner cell mass of the mouse blastocyst grows deeply into the blastocele. As a result, the mouse embryo becomes cup-shaped and is referred to as the egg simulacrum, Fig 7.36. The inner cell mass becomes bilaminar and the epiblast lines the inner surface of the cup. The trophoblast cells near the inner cell mass, polar trophectoderm, also proliferate forming the ectopbnental cone, and this further elongates the embryo, Fig 736. The shape change makes it more challenging to follow murine gastrulation and organogenesis than these processes in amphibian and avian embryos. At about 6.5 days after fertilization, Thyler stage 9, the primitive streak forms in the caudal region of the epiblast, Fig 7.36, 7.37. The primitive groove is distinct by day 7, Thyler stage 10. At the rostral end of the primitive streak is the NMK, which is located at the bottom of the cup-shaped embryo. The mouse node is analogous to Henson's node of Avian embryos in that it is a mammalian organizer. As murine gastrulation proceeds, the node becomes located more caudally, on the side of the cup. Human gastrulation. Similar to avian and porcine embryos, the human embryo is flat and disc-shaped, Fig 7.38. The embryonic disc shown in figures 7.38 and 7.39 appears sandwiched between TV.RO bubbles made of extraembryonic membranes. The bubble dorsal to RHE embryonic disc is the amnion, and RAR ventral to it is the umifo-LNIDC, yolk sac. Since the human yolk sac does not carry yolk, the term umbilical vesicle is preferred. The primitive streak appears in the bilaminar embryonic disc by day 17, Carnegie stage 6. At the rostral end of the primitive streak is the primitive node, Fig 7.38. Morphogenetic cell movements of gastrulation convert the bilaminar embryo into a trilaminar embryo with the cells in their proper positions, Fig 7.38 to 40. One of the most intriguing features that distinguish chordates from other animals is the presence of the hollow nerve cord. In vertebrates, the nerve cord expands into the central nervous system, brain and spinal cord, with the brain cranial and spinal cord caudal. How does the nerve cord form? Neurulation is the process in which the rudiment of the nerve cord, called the neural tube, forms and separates from the overlying surface ectoderm. The neural tube is a hollow cylinder of Nker.1 ectod cream cell, Nkurokpithcleum. An embryo undergoing neurulation is called Nkural.I. Overview of neurulation. The ectoderm of the neurula differentiates into three major cell types: 1. Skink. Cream, epidermis, future skin; 2. Neural ectod cream, future central nervous system; and neural CRCT, forms part of the peripheral nervous system and several non-neuronal cell types. This chapter focuses on the neural ectoderm and the process of neurulation, by which it develops into the neural tube. There are two types of neurulation: 1. Primary neurulation, in which the neural ectoderm folds to form a hollow neural tube, and 2. Secondary neurulation, in which mesenchymal cells aggregate to form a solid neural rod, medullary rod, that hollows out to form the neural cube. In frogs, birds, and humans, most of the neural tube forms by primary neurulation, and only the most caudal region develops by secondary neurulation. Caudate embryos that undergo primary neurulation use a general pattern, Fig 8.1. First evidence of neurulation appears on the embryo's dorsal side in ectoderm lying above the chordum mesoderm. The chordum mesoderm is a group of condensed mesoderm cells that lie on the dorsal side of the embryo, between the ectoderm and endoderm of the embryonic gut. The chordum mesoderm differentiates into the notochord, a longitudinal, rod-like structure that runs most of the length of the embryo between the digestive tube and the nerve cord. In many species, the chordum mesoderm/notochord produces inductive signals that allow the overlying ectoderm to differentiate into neural ectoderm. The neural ectoderm cells elongate into a columnar shape, forming a flat, longitudinal plate of cells called the neural plate. The lateral ends of the neural plate rise up into neural folds, giving the neural plate a U or V-shaped appearance. Between the neural folds is a depression, the neural groove. The raised neural folds move toward each other and fuse to form a hollow, cylindrical neural tube that becomes covered in skin ectoderm. The neural tube's cavity is the nurmicle. The cranial and caudal tips of the neural tube close last, and their open ends are called the cranial neuropore, rostral neuropore, anterior neuropore, and caudal neuropore, posterior neuropore, respectively. Neurulation in model organisms. Since there are species differences in exactly how neurulation is achieved, this chapter examines neurulation in several model organisms. Further differentiation of the neural tube into the central nervous system will be covered in Chapter 9. During neurulation, the embryo also develops non-neuronal structures, i.e., gut, heart, chylome, that are primed to form as a result of cell movements during gastrulation. For clarity, these non-neuronal structures will be discussed briefly here as necessary, in more detail in Chapter 9, and more specifically for the frog, Chapter 10, bird, Chapter 11, pig, Chapter 12, and human, Chapter 13. Cephalocorded neurulation. The lancelet, Amphioxus, branchiostoma, is a simple organism to study the events of neurulation, Figs 1.6, 17, Table A6. The early neurula elongates and becomes ovoid and flattened on the dorsal side, forming the neural plate, which forms a shallow groove directly above the chorda mesoderm, Fig 8.2. At this stage, the chorda mesoderm forms the roof of the archenteron. Soon the skin ectoderm at the edges of the neural plate moves across the dorsal midline and fuses. This causes the underlying neural plate to form a V shape, and the ectoderm to completely cover the dorsal side of the neural plate. The folds of the neural plate eventually fuse from posterior to anterior to create a neural tube that remains open anteriorly for a short time as the anterior neuropore, and posteriorly as the Kirchenkrieg canal, which is temporarily connected with the gut. The lancelet, B. floridi, neurula hatches from the fertilization envelope by 10 hours after fertilization at 25 degrees Celsius, Table A.6. When its mouth is open to the environment, the embryo is called a larva. Teleost neurulation. Unlike vertebrates that undergo primary neurulation, fish undergo secondary neurulation. The neural tube develops from a solid cord of condensed cells, which later hollows out, Figs 8.6, 8.7. During gastrulation, neural ectoderm cells migrate into the dorsal midline of the epiblast of the embryonic shield, and elongate to form the neural plate. Aggregation of neural plate cells forms the neural keel, a thickened solid cord of cells in the mid-dorsal axis of the embryonic shield. The neural keel detaches from the overlying skin ectoderm and sinks toward the underlying chorda mesoderm, Fig 8.7. The detached neural keel eventually develops a central canal, thus transforming the neural keel into a hollow neural tube. Anteriorly, the neural tube dilates and gives rise to the brain and optic sides, future eyes. Posteriorly, it forms the spinal cord, Figs 8.8 to 8.10. While the optic vesicles are forming, the yolk plug in the blastopore is covered with ectoderm by the apibolic movements of the germ ring. This signals completion of gastrulation. Note that before gastrulation is complete, neurulation is well underway in fish embryos. Amphibian neurulation. The majority of the amphibian neural tube forms by primary neurulation. The neurocord sends inductive signals to the overlying dorsal ectoderm cells, which respond by becoming columnar in shape. The patch of columnar ectoderm cells will become the future central nervous system and is first visible as the pear-shaped neural plate, Fig 8.11. The wider anterior end will form the brain, and the narrow posterior end will develop into the spinal cord. At the mid-neurula stage, the neural plate quickly elongates and simultaneously its lateral edges rise up above the ectoderm as neural folds that surround the neural groove. By the late neurula stage, the neural folds fuse in the midline to form the hollow neural tube and neurocoel. While the neural tube forms, it sinks into the embryo toward the notochord and becomes covered with epidermis. The most posterior region of the spinal cord forms by secondary neurulation. Avian neurulation. The epiblast is the site of neurulation in the avian embryo. While the neural ectoderm undergoes neurulation, the embryo also develops mesoderm and endoderm derivatives. The avian neurula is not pigmented and is relatively transparent as compared to the frog neurula. Thus, mesoderm and endoderm derived structures lying ventral to the apiblast are visible not only in sections, but also in whole mounts, in addition to neuronal structures. Neurulation begins in the cranial region of the chick embryo and gradually progresses caudally, Fig 8.24. Neurulation does not occur in the avian embryo all at once. While the neurulus cranial region undergoes neurulation, neural folds are resexable, its caudal region has just begun gastrulation, primitive streak is present. Therefore, both Gastrulation and neurulation occur simultaneously in different regions of the chick embryo. Because the blastoderm forms within the hen's oviduct, the extent of development can vary widely at the time the egg is laid. Development stops, due to the cooler temperature outside the hen body, until the egg is incubated. Significant variations in development occur by 24 hours of incubation. Thus referring to incubation time is not a reliable indication of progress of early embryonic development. It is more accurate to relate neural development to the number of paired somics, paired blocks of mesoderm to the right and left of the notochord. Actually, the word pair is not included in the somite description, so that a 5-somite embryo is understood to have 5 pairs of somites. Or 10 individual somites, 5 lying to the right of the notochord, and 5 to its left. The staging system of Hamburger and Hamilton, Table A9, uses the number of somites to distinguish between early developmental stages, from about 20 to 50 hours of incubation. Stage 7 has 1 somite pair. A new stage is assigned every 3 new somite pairs that have formed, hence, stage 8 has 4 somite pairs. For in-between stages, plus and minus signs are used. For example, an embryo with 2 somite pairs is a stage 7 plus, and one with 3 somite pairs is a stage 8-. Before studying whole mounts and sections of the embryo, it is advantageous to understand the location of neuronal and non-neuronal structures that will be seen simultaneously in the transparent chick embryo. Figure 8.25 shows the cranial region of a 6-somite, 24-hour, chick embryo in whole mount, sagittal section, and transverse section. Note that in sections, the neural folds and neural tube are dorsal, and the foregut is ventral with the rod-shaped notochord lying in between, whereas these structures appear to be in the same focal plane in the transparent, thin whole mount. Development of the avian nervous system depends on the notochord, Fig 8.24. During gastrulation in the cranial epiblast, the primitive streak begins to regress and Henson's node moves caudally, leaving behind the ingressed neurochord cells. For example, Henson's node is located a little over 1/2 the length of the 2-somite embryo at the 20-22-hour stage. Ver has regressed to the caudal 1/3 or 1/4 by the 24-hour stage, Figs 8.24, 8.26, 8.29, 8.33. The prospective notochord cells are first visible in the 18-hom chick embryo as the HCAL process, Figs 7.28 to 7.33, 8.24. The ectoderm overlying, dorsal to, the head process develops into a broad neural plate of thickened neural ectoderm, Fig 7.34, 8.24. The neural plate is distinguished from the primitive streak by the presence of the notochord ventral to the neural ectoderm, Fig 7.28 to 7.33, 8.24. As neurulation progresses, the neural plate narrows and lengthens by convergent extension. The neural plate bends ventrally in the midline in 2-somite chick embryos, 20-22-hour, to form the neural folds that are separated by the neural groove, Figs 8.24, 8.26 to 8.28. In the whole mount, neural folds appear as 2 parallel dark lines in the cranial dorsal midline of the embryo. In transverse sections of the embryo's anterior, the neural folds are U or V-shaped areas of thickened neural ectoderm. As transverse sections of 21-hour chick embryos are traced caudally, the neural folds diverge as they flatten into the neural plate, Fig 8.28 bg. This also holds true for the 24-hour chick embryo, Fig 8.371j. The neural folds overlay the somites, but are obscured by them and are more readily visible in transverse sections. Figures 8.29 to 8.37 show a range of development in different chicken eggs incubated for 24 hours. The embryos have 5 to 7 somite pairs. The elevated neural folds will either soon meet in the dorsal midline and fuse to form the neural tube in the 5-somite embryo, Fig 8.29, or the neural tube has already formed in the cranial region of the 6-somite embryo, Figs 8.30 to 8.36. At the rostral tip of the neural tube, the neural folds are unfused, leaving a temporary opening, the cranial neuropore, Figs 8.29 to 8.33, that closes by 33 hours, stage 10. By 48 hours, stage 13, all of the neural folds have fused, indicating that primary neuralarian is complete. The notochord is visible directly ventral to the neural ectoderm of the neural folds and tube. Note that the notochord does not develop into the brain and spinal cord, but is located ventral to these structures. Mammalian neurulation. Neurulation in mammals is very similar to avian neurulation, except that it occurs more slowly. While neurulation begins by the first day after fertilization in birds, it does not begin for at least a week or more in many mammals. Tables A10, A11. Both gastrulation and neurulation occur simultaneously in the trilaminar mammalian embryo, Fig 8.38. The mesoderm-derived ectochordal pros, analogous to avian head process, induces the overlying ectoderm of the epiblast cranial to the primitive node to become a thickened neural plate. Thus the neural plate first appears cranial to the primitive streak and elongates both cranially and caudally as the primitive streak regresses and the embryo grows. The neural plate is broad cranially and narrow caudally. The broad region becomes the brain, and the narrow portion develops into the spinal cord. The cranial neural plate exhibits swellings and constrictions that will later differentiate into different brain regions. The pig neurula is flat and oval when the neural plate develops, and becomes shaped like the sole of a shoe when neural folds appear, Fig 8.38. The neural folds and neural groove form about day 13, Table A11. The notochord lies ventral to the thickened neural ectoderm, which rises up to form neural folds that fuse when about 6 somite pairs have formed, Figs 8.39 to 8.41. By -15 days, the neural folds fuse except at the cranial and caudal neuropores, which close later. The cranial neuropore closes on day 16 and the caudal neuropore closes on day 17. During neurulation, the primitive streak regresses. The mouse neurula is cup-shaped and appears bent back on itself. The notochord lies ventral to the thickened neural ectoderm. The neural plate folds along its midline, forming the elevated neural folds and neural groove, Table A10. The neural folds first close at the level of the 4th and 5th somites. Closure of the neural folds to form the neural tube progresses rostrally and caudally. The cranial and caudal neuropores close last. The cranial neuropore closes at about 8.5 to 9 days, thylar stage 14, and the caudal neuropore closes by day 9.75 to 10.25, thylar stage 16. Human neurulation. In humans, first sign of neurulation is the shallow neural groove in the thick neural ectoderm, which appears during Carnegie stage 8, Table 8.1, Figs 8.47, 8.48. The lateral edges of the neural plate elevate to form neural folds as the neural plate bends in the midline, forming the neural groove. During Carnegie stage 9, the neural groove deepens, Table 8.1, Figs 8.49, 8.50. During Carnegie stage 10, at the level between the future brain and spinal cord, the neural folds begin to fuse to form the hollow neural tube with its space called the neural canal, Table 8.1, Figs 8.51, 8.52. The two open ends of the neural tube are the cranial, rostral, neuropore, closes at Carnegie stage 11, day 29, and the caudal neuropore, closes at Carnegie stage 12, day 30. Unlike birds, in which neural tube closure begins at the level of the future midbrain and then zips closed in both directions, human neural tube closure is initiated at several sites along the midline of the neural plate. Failure of the neural folds to fuse at these sites results in different neural tube defects, such as anencephaly cranially and spina bifida caudally. After normal neural tube closure, the hollow neural rube is located dorsally in the embryo, and is covered with skin ectoderm dorsally. The notochord lies ventral to the neural tube. The notochord eventually becomes surrounded by bone of the vertebrae, but persists between the intervertebral discs as the nucleus pulposus in the fetus, newborn, and young children. It is unclear whether the neurocord cells persist in the adult. The lower lumbar portion of the spinal cord forms by secondary neurulation.